Thank you, everybody. Greetings from Nairobi, Kenya. Welcome for the 5th seminar in the series Crop to End Hunger, Case Studies in Africa and Beyond, supporting the CGIAR partners through genotyping services. Warm regards to all. So in our previous seminar, we had Charles Nellimore that presented us interesting updates on West African maize land races and how these related to drought and heat tolerance. We had a lot of feedback, good feedback during and after the seminar, so hopefully today we will have the same. And today we are presenting a senior scientist from CGIAR India, Dr. Salesh Sood, to talk about potatoes and their breeding program. And as in other sessions, we are very pleased to welcome Dr. Lenin Muzundir. He is the African NARS breeding optimization specialist and working across crops and NARS in Africa and Asia. He has a very strong breeding background on the public and private sector, and he will introduce Salesh. Thank you, Lenin, for accepting this invitation in the last moment. Thank you really very much. Please go ahead. You are muted, Lenin. Kindly unmute you.
Okay. Thanks, Anna. I was just talking to myself. You not unmuted me. Yeah, all right. That's fine. Thanks, everyone, for joining this seminar. And it's an honor to be introducing our guest speaker today. And our guest speaker is Dr. Salesh Sood, who is a senior plant breeding and geneticist at ICA Center, Potato Research in India. A rich history, over 17 years in plant breeding. And he is a holder of a PhD with significant contribution towards potato and millet varieties and technologies in India. Dr. Sood has created 8 millet varieties, 2 potato varieties, and 8 genetic stocks that are currently enriching the breeding programs in India. He excels in conventional and molecular breeding methods, mainly focusing or specializing in marker-assisted selection and genome-wide association studies and genomic selection. His expertise includes germplasm characterization, field experimentation, statistical methods, and managing external funded projects. Dr. Sood has authored over 70 peer-reviewed publications and conducted postdoctoral research on genomic selection at La Trobe University, Melbourne, Australia. He received prestigious awards from institutions and societies, including National Academy of Agricultural Sciences, India Associationship, Endeavour Research Fellowship, and the Fellowship of the Iranian Society of Genetics and Plant Breeding. So before we go into him presenting, I would like also to give a background on on today's presentation, where he's going to present on potato cultivation in Africa. And for background, potato cultivation in Africa has steadily grown, playing an increasingly significant role in the continent's agricultural landscape. While the potato is not native to Africa, it has adapted well to various African climates and has become a vital staple crop in many regions. One of the key reasons behind the rise in potato cultivation is its versatility, adaptability to diverse environments, from highland areas with cooler temperatures to lowland regions with warmer climates. In East African countries, for example, Kenya, Ethiopia, and Uganda, these have witnessed substantial increase in potato production, making it the crop of choice in the current status. These countries benefit from their high altitude regions, which provide optimum growing conditions for potatoes. Smallholder farmers have embraced potato farming due to its relative short growing season and the potential for higher yields relative or in comparison to traditional staple foods like maize and cassava. Additionally, potato nutritional value, rich in carbohydrates, vitamins, and minerals, makes it a valuable crop in efforts to combat malnutrition and improve food security in particular here in Africa. Egypt is a notable producer in North Africa with extensive cultivation in the Nile Delta region. Egyptian farmers have adopted modern farming practices, technology to boost potato yields and meet the growing domestic demands for this versatile crop. Potato cultivation in Africa, however, faces a barrage or a series of challenges such as pest and disease management, access to quality seeds, and post-harvest losses. Collaborative efforts between government, non-governmental organizations, and international organizations aim to address these issues and promote sustainable potato farming practice, ultimately contributing to food security and economic development across the continent. In India, potato, even if not indigenous to the country, it is a vital component of its agricultural landscape. It plays a crucial role in its food security and economy. Northern states like Uttar Pradesh, West Bengal, Punjab, and Bihar are the major potato producing regions in India. These states benefit from fertile soils, diverse weather patterns, and supporting winter and summer and summer potato crops. I will not say much, but I will open the floor for Dr. Salej Sood to present or to give out the presentation that he's going to give us. Thank you very much. And Dr. Sood, please go ahead with the presentation.
Thank you, Lenin. Thank you for a nice presentation and nice introduction of myself. Thank you, Anna. Greetings to all who have joined from different parts of the world to attend this seminar. Let me share my presentation. Is it visible?
Yes, just put in a presentation mode, Salej, please.
Okay.
Yes, it's okay.
Yeah, nice. So today's topic is accelerated. Just a minute. Let me. Anna, can you look after the people who are coming? Can you admit them so that I can focus on presentation?
Yes, don't worry, I will manage that. Thank you.
Okay, thank you. So today's topic is accelerated genetic gain in potato breeding through genomics-led breeding. And in this, I'll be the basic aspect of the presentation will be a brief introduction about the crop and its scenario in India, the global scenario, and then the work which I have done or our team at CGIAR has done along with the excellence in breeding team. My slides are not moving, Anna. Okay, I have to move, I think, with the cursor. Thank you. So a brief introduction about the potato. So potato, everybody knows it's a crop of Andean region of Peru. It's domesticated around 8,000 years ago. And now it has become the third most important food crop after rice and wheat in terms of human consumption. And the Solanum genus under which this potato comes, this has Petota section under which this potato crop comes, and it has around more than 100 tuber-bearing species. And the important cultivated species of potato, everybody knows, is Solanum tuberosum. But it has two groups. One is Chilotinum group and another one is Indigenum group. So the Chilotinum group is long day adapted and the Indigenum group is short day adapted. And apart from this, there is another group that is Neotuberosum, which is supposed to have been bred from this Chilotinum and Indigenum hybridization. As we know that the potato genetics is complex actually because of its tetraploidy and vegetative propagation and the heterozygosity of the clones due to the vegetative propagation of the potato. And if we see this figure, if we compare with diploid crop like rice and this tetraploid potato, we can see that to select a particular individual, we need to generate more number of progenies. And this is the case of a single locus. So if we compare rice with potato, in rice, we need only the 4 individuals if one loci is segregating. And in case of potato, we need around 36 individuals to select a particular phenotype. This is the journey of potato from its center of origin. So this shows that how the potato has moved across the globe. And in India, it came from UK and Portugal in the 17th century. This is the global scenario of potato. So if we see here, the world production, as per the FAO stat, recent FAO stat, it's 376 million tons. And Asia and Europe, they together contribute about 80% of the potato production. The major producing country is China, which is followed by India, and then Ukraine, USA, and rest of the countries. And about 50% of the potato is consumed as fresh food. If you see the Indian scenario, in India, the potato is grown as a subtropical crop. I think everybody knows that potato is a temperate crop when it originated actually, and it was domesticated initially. But in India, it is grown as a subtropical crop majorly, and now it has moved to even the tropical regions also. It's a predominant vegetable in India now. It's mainly consumed as fresh food, and the processing sector is growing in India recently. In last 10 to 20 years, around 10% of the production is going for the processing sector. But we have inadequate storage capacities. Although the production has increased, but the storage capacity is inadequate. And India is producing about 53 million tons of potato from around 2.2 million hectare area. And the average productivity in India is around 25 tons per hectare. Out of this, around 4.5 million tons, that is around 8.5% of the produce is used as a seed, and 10% of the produce is processed by the processing sector, and around 0.5 million tons is exported. This is the figure of 2022. And as Lenin also told about the post-harvest losses in Africa, in India also the post-harvest losses are around 20% of the total produce. These are the agroecological zones where the potato is grown in the country. So you can see here that potato is grown right from the northern hills, eastern hills, central plains, northern plains, and even in the southern hills and in the plateau region also. This is about the organized research in India on potato. So the CPRI started in the year 1949. It was first started in the plains in Bihar, but later it was shifted to the northern hills, that is in Shimla, because of the hybridization requirement, because potato requires long day conditions for flowering. So to breed new varieties, the headquarters was shifted to Shimla. And the first indigenous variety from India was released in the year 1958. Next are the things which are related to seed production. So I'll not go into the details. And these are the centers or the organized research centers of CPRI in India. So we have centers all around the country, whether it's our own research center. We have 6 research centers which are of CPRI. And apart from this, we have a coordinated, all India coordinated research project whose headquarters is also at CPRI Shimla. And we have 25 centers under this AICRP program, out of which 7 centers are our own centers, that is CPRI centers, Central Potato Research Institute centers, and 17 centers are state agriculture university-based centers. And these centers are basically to evaluate or to conduct the multi-location testings of advanced clones. These are the varieties which have been bred in India, and you can see here the varieties as per the duration, the number of varieties which are bred in early category. These are the 12 varieties. And in medium maturity duration, we have 48 varieties. In the late maturity duration, we have 10 varieties. And then the categorization is based on the agroecologies, that is for northern hills, northern plains, plateau region, Eastern Hills, and Southern Hills. So nowadays, our major focus is on processing varieties and developing varieties resilient to climate change, that is heat-tolerant varieties, or spreading our potato cultivation to the tropical areas or the non-conventional areas. Coming to the work which we have done with the support of Excellence in Breeding team, there are people I can see here who are in this seminar. Dr. Rajaguru is there, and even Gary Atlin I can see. So I'll be presenting the work which we did with the support of Excellence in Breeding. So these are the three parts. First, we evaluated or assessed the genetic gain which we have achieved through potato breeding in India. And then we developed some low density markers or the CASP markers for forward breeding in potato. And we are also doing this mid density genotyping using the DARTAG panel, which have been specifically designed for genomic selection in potato. So first I'll tell about the genetic gain which we have achieved through potato breeding in India from 1968 to 2012. So here we use the ARA trial methodology. There are two methodologies which can be used for getting the realized genetic gain. And we used here the ARA trial methodology. In this methodology, we evaluated 22 potato varieties which have been released from 1968 to 2012 in India. And these trials were conducted from 2014-15 to 2016-17 for 3 consecutive years in different locations. And this data was divided into 4 zones, that is Northwestern Plains, West Central Plains, Eastern Plains, and the Hill region. So here the number of varieties are given in this table, which have been evaluated in each zone, and then the location and the relative genetic gain, which we have observed for different traits. So these are the 3 traits which we evaluated here: total tuber yield, marketable tuber yield, and the dry matter. So if we see here, the maximum annual genetic gain which we observed in case of potato breeding in India was 0.404% annual gain, that too in West Central Plains. And for rest of the regions, for total tuber yield and marketable tuber yield, the genetic gain was lower than this. And if we see the dry matter case or the quality parameter in potato, the genetic gain was low all across the zones, except in Hill region where the crop duration is more. So probably that was the region that we got higher genetic gain for dry matter here. And this is the regression of the mean of different traits of varieties on the year of release. So we can see the regression slope here. So for the first one is dry matter, the second one is marketable yield, and the third one is total yield. So we can see here for marketable tuber yield and the total tuber yield, the recent varieties have shown the genetic gain. But in case of dry matter, there is no genetic gain if we compare from 1968 to 2012, except in the Hill region. So this shows that the genetic gain in potato breeding is low as compared to the other crops, and as well as the targets which have been set by the CG, that is the 1% annual genetic gain in all the crops. Coming to the various biotic stresses which hamper potato quality and quantity. So among these, there are a number of biotic stresses, but late blight and viruses and PCN, these are the 3 important biotic constraints which affect the potato production and productivity. And among these also, the late blight is the most prevalent disease which affect the potato production in all areas and in all the hilly regions, or we say the plateau regions of India. And among viruses, PBY is the major virus which affect the potato production mostly in the plains, northern plains, in the central plains. And PCN again is a temperate pest which affect the potato production in the hilly areas all across the globe, or the temperate areas. And For screening, if we say that we have to screen for these biotic stresses, it's very difficult in potato breeding, because in potato breeding, when you cross two genotypes or two clones, you get number or million of TPS. And when this TPS is raised in the form of seedlings, or you get the clones, so you have to screen all the clones to get or to forward your selected clones for that particular or desirable trait. So marker-assisted selection is the best approach in that case. And although there are markers available and are being used in potato breeding for long, maybe for 20 to 30 years, but if we see the CASP markers, they are the high throughput markers, and they can be used for screening of large number of germplasm. So for this, we generated CASP markers for different biotic stresses, that is for late blight, PBY, that is potato virus bite, potato cyst nematode, plant maturity and maturity corrected resistance, chip color, and common scab. And among these, for late blight, we had markers, some validated markers from SIP, and we also developed our own markers. Around 2 to 3 markers we got from SIP, and rest of the markers we generated by our own, based on our GWAS studies and also the literature-based search. And these markers were then validated in a set of 1447 potato accessions, which was used for screening for different years, or this was the historical data on these 3 traits, that is late blight, PBY, and PCN. For rest of the traits, we are generating, or we are still evaluating this genotype data and phenotype data for association. This is the procedure of our CASP assay development and evaluation. So here I've just shown that how we prepare the samples, and then these are sent to the Intertech lab in India, and then the data analysis part. So the first part is the leaf collection. So we generally collect the leaf from 4 to 6 leaf in the potato plant, which are grown in the pots or in the fields. And then 2 leaf discs are taken with the help of this puncher, which is shown here. And then these samples are dropped as per the layout, which we fix in these deep bell plates. Then the samples are dried in oven at 40 degrees centigrade for 48 hours. And then these samples are sent to the Intertech lab. So the arrows which I've shown here, the green arrows, these samples are prepared in our lab. And after that, from this step, the yellow arrow, these samples are once sent to the Intertech lab. They do the high throughput DNA extraction. They do the CASP PCR analysis, and they send us the results in the form of fluorescence signal detection. And then the data analysis and validation part is done at our end. So these are the 9 markers out of all the markers which I've shown in the previous table. These 9 markers, they were validated in potato with the help of these phenotype data. And out of these markers, the 3 markers are for late blight, 5 markers are for PBY, and 1 marker is for PCN. And in this table, we can see the resistant allele, the susceptible allele, and the gene associated with these SNPs. And these are the amplification results and the allele calls for different SNPs. So we can see here we found polymorphism for all these markers. We can see all the 3 type of classes: the favorable allele, the heterozygote, and the unfavorable allele in all these markers or all these CASP assays. And this is the validation part. I'm just showing here for 1 trait, that is for late blight. We use these 3 markers, or 3 markers were validated. And in these 3 markers, we can see here that this table on the right-hand side, we can see here the resistant, moderately resistant, and susceptible accessions based on the phenotype data. So in the favorable, if we see the first CASP assay, that is for R2 gene, for the favorable allele, there were 3 resistant accessions, 4 moderately resistant accessions, and there was none in the susceptible category. So it shows that the markers worked well. But if we see the other two markers, the R8QTL 2 caspase, for the favorable allele, for the first caspase for R8QTL, in the resistant category, none of the accessions was found. In the moderately resistant categories, 2 accessions were found, whereas in the susceptible category, none of the accession was there. Similarly, for the second caspase for R8QTL, there were 2 accessions which were in the resistant category for the favorable allele, 4 in the moderate resistant category, and we also found 1 in the susceptible category. And this is bound to happen actually, because in potato, there are a number of genes which have been found for late blight resistance, and the pathogen also is highly virulent. It keeps on mutating, and number of genotypes are being number of genes are being identified for resistance. Coming to the results of these CASP assays in the form of scatter plots, we can see here the clear segregation of the favorable allele, the unfavorable allele, and the heterozygote classes. And we also developed CASP markers for self-compatibility screening, because nowadays, if you see in potato, there is a new approach, new breeding approach, that is diploid hybrid breeding approach, which is picking up, which is a TPS-based propagation approach, where the concept is to develop the inbred lines at the diploid level, and then to exploit the heterosis in potato. So here we can see that we developed 10 CASP assays from this SLI gene, which is present on the 12th chromosome of potato, this is a study which has been conducted by Clot and co-workers. And from their study, we picked up these markers, and then we ran these markers in our germplasm. And we found that there was clear segregation of self-compatible and self-incompatible accessions. So here you can see the allele call results in the form of these segregation plots. And if we see the data, when it was compared with the phenotype data, the genotype data of CASP assays, we did the chi-square, we applied the chi-square test, we found that except the one CASP assay, which showed the significant results, all other CASP assays, they showed non-significant results, which showed that the phenotype data was perfect with the genotype data for self-compatibility screening. That is, these 9 markers, these 9 CASP assays can be used for screening for self-compatibility in diploid potatoes. Similarly, we also developed 100 CASP assays for homozygosity testing at the diploid level, since I already told that at the diploid level, the purpose or the approach is to develop inbred lines first by repeated selfing of the initial founder heterozygous lines. So for this reason, to check the homozygosity in different generations, we developed this new 100 CASP assays, and which are distributed across the genome in potato. And these SNP sequences were taken from a previous study, which was done by Anita Kumari and co-workers. And these SNPs are distributed on all the 12 chromosomes of potato. You can see the distribution of these SNPs here in this figure, which are distributed across the 12 chromosomes in potato. And then when we check the homozygosity percentage based on these 100 CASP assays, we found in our diploid material that when we checked our founder diploid lines, the minimum homozygosity was 20% and the maximum was 78%. Similarly, when we went down with each generation of selfing, with the increase in selfing generation, the homozygosity percentage increased. The minimum level also and the maximum level also. And if you see the average homozygosity also in these clones or these lines, that also increased with each selfing generation, which was evaluated with these 100 CASP assays. And here we can see in this figure that the cells with green color, they shows the homozygosity at that particular loci, and the cells with yellow phone, they shows the heterozygosity at that particular loci. So if we see here, the first top figure, these are the heterozygous founder lines. Here the heterozygosity is high. The yellow cells are more. When we go down with one generation of selfing, the green color cells are more. It means the homozygosity has increased. And same is the case in after 2 generations of selfing. And if we see after 3 generations of selfing, the homozygosity reaches to about 88%. Coming to the next part, that is the integrating genomic selection in our potato breeding program. So for this, we first what we did is we used this mid-density genotyping panel for genotyping our potato clones in the processing breeding program. And in this although they are telling that it's a 2.1K panel, but we got around 2,500 SNPs, and out of these 2,500 SNPs, the 60 loci which has read depth of less than 10, we removed those loci, and rest of the SNPs or the rest of the data was used for associating with the phenotype data. And the phenotype data, it was generated in 2 locations, although we also tried to use the historical data, but the number was quite less. It was in around 20 or 30 clones were there. So that was the reason that we tried to develop a new data set on phenotype of these processing traits to use this GS model and use the integrate genomic selection in potato breeding program. And this is the panel. This is the distribution of SNPs in this panel. That is a 2.1K panel in potato. And we can see this is a good panel and specifically designed for genomic selection in potato. It has 2 to 3 SNPs per centimorgan. The genotyping cost is low. As of now, the cost is around $12 per sample. And there is every possibility to include any genic marker in this panel. And when we use the GBLUP model based on the available data, this is a preliminary data, I would say, as of now, because the number is less, and we have included the parents also in this data, which we are not supposed to do in the breeding program for genomic selection, but just to test this, we included the parental lines also. And when we check the prediction accuracy, which is actually the correlation between our observed phenotype data along with the GEBB values, that is genomic estimated breeding values. And what we did here is actually we masked 50 individuals for 50 replications from our phenotype data, and then we evaluated those 50 individuals for 50 replication to estimate the GEBB's values, and those GEBB values were then correlated with the observed phenotype value. And this correlation, it shows the observed phenotype value for 50 replication. So this value is actually for the range of 1 replication to the 50th replication. And here we can see the prediction accuracy for different traits. And we can see the one which I've highlighted with red font, the prediction accuracy were good for these traits. And the one which are black in color, the prediction accuracy was quite low for these traits. But for most of the traits, the prediction accuracy was good. So this shows that this is although a preliminary results, but this shows that this genomic selection can be integrated in potato breeding program, provided we develop robust data on our traits for which we are looking to integrate in our breeding program. And these are the box plots for this genomic prediction accuracy. So here, the point here to show this box plot is just to show that the traits for which we observed high prediction accuracy, and there are traits for which we got low prediction accuracy. So we need to improve upon these traits, or we need to have more data, robust data for these traits, so that we can then predict, or we can see whether our prediction accuracy is increasing with the more data or not. And this is the prediction bias for the traits for which we use this genomic selection. And we can see here TT by is total tuber yield. So our prediction was almost reliable. We can say it was good and reliable. For specific gravity also, the prediction was reliable. Similar was the case for the tuber shape. But if we see the flesh color, some of the data points, they were away from the regression line. So although the prediction accuracy, if we see in the table, it was good, but the reliability was low. And based on our results, which we have achieved, or by comparing our conventional breeding program, where we can incorporate this genomic selection in our breeding program is based on these last 3 years data, we found that in conventional breeding program, what we are doing is the first year, we are crossing the genotypes or the clones. Then second year, we are raising the glasshouse seedlings. We are getting the F1 clones, F1C1 clones. Those F1C1 clones in potato, actually, each individual is a new clone. So you need to multiply the tubers till you can evaluate them in replicated trials. So for year 3 and 4, the tuber multiplication is done, and the data is generally recorded based on the visual preference. So that is breeder's visual preference. And from year 5 to 7, the replicated trials are done, where the actual data on yield, quality, and biotic stresses are generated. And then in the 8th to 9th year, the station trials, yield, quality, and disease resistance is again carried out. And from year 10 to 12, the multi-location trials are conducted across the locations to see whether any clone is performing good, and that is released as a new cultivar. This is our existing system in potato. So with the genomic selection breeding scheme, we proposed based on the results that when we are doing the crossing in the first year, second year, our glasshouse seedlings, we are generating clones. And then in the F1C2 generation, the field seedlings are there. Then in F1C2 itself, we genotype the F1C2 clones and then predict the, we use the genomic prediction model here and see which clones are showing a higher GEBB for a particular trait of interest. And those clones are then taken back into the crossing block based on the GEBB's values. And then this recurrent selection or cyclic selection goes on to improve the genetic gain in potato breeding. And by applying this, the time period is actually reduced, or it is shortened a lot. And the genetic gain is also increased because we are increasing, or we are actually accumulating the desirable alleles for our traits of interest. So coming to the summary of our work, so we found based on these studies that genetic gain in potato is low. It's not only low in India, but when I saw the literature or we surveyed the literature, it's low across the regions in case of potato. And that is possibly due to its complex inheritance and the clonal multiplication. So that could be the reason that the genetic gain is low in potato breeding. And we found that CASP assays demonstrate the usefulness in high throughput, cost-effective marker-assisted selection for biotic stress traits, but that also need to be done with precaution, because initial evaluation, preliminary evaluation can be done with CASP assays for biotic stress traits. But when the number of clones are less, we need to use the phenotypic selection for those particular biotic stress traits. The genomic prediction accuracy is moderate to high for most of the traits in potato breeding, but that again, we need to generate more data in the breeding program and for more number of locations so that we can integrate genomic selection in our breeding program, as I showed in my previous slide. That is, we can integrate it in the second clonal generation, that is in the F1C2 generation. Again, the same thing, that the robust and accurate data could improve the prediction accuracy of genomic selection. And once we have the robust and accurate data, that will be the time when we can actually depend upon GEBB's values to bring back the individuals in our crossing block or improve the parental lines. Genomic selection, it shows the promise for enhanced genetic gain in potato breeding, and it will take some time, but definitely it will be integrated in our breeding programs. These are some of the publications which emerged out of our work with the EIB team. And first we evaluated, as I showed earlier, that what is the genetic gain in potato breeding in India. And then we developed some CASP markers and we validated those CASP markers for 3 biotic stress traits. And we also did some digitization work in potato breeding program where the multi-location trial data is being digitized. And then we also did some genome-wide association studies, and genomic prediction it's going on, and we are hoping that it will be integrated in soon in our breeding program. With this, I end my presentation, and I would like to thank and acknowledge our DDG Horticulture, our director at CPRA Shimla, my CPRA colleagues who generated data, the ICR BMGF grant, Dr. A.K. Singh, who is the coordinator of this ICR BMGF project, and Dr. T. Napoleon, who was genotyping lead for this project, Dr. C. Bhardwaj, and from EIB team, Dr. Anna, Dr. Abhishek Rathore, Rajguru Bohar, and many more. Gary's here, and it was the first time when we interacted with Gary when this project was started at ICRISAT. So I would like to thank Gary also, and Eduardo also. Thank you one and all.
Thank you, Salesh. Thank you very much for the great presentation and good data. So now we open floor for questions. Gary, I saw that you already have 3 comments. Please, I can unmute all the participants. Dorcas also have a question, but we can start by Gary. Gary, can you unmute yourself and you make the comments and questions, please?
Sure. Thank you very much. Very nice presentation, Salesh. Lots of great progress from that team. Just a couple of thoughts about genomic selection. We're really not suggesting that genomic selection be used mainly to identify good potential varieties from the early stages. It's not very helpful for that, really, except in fairly limited situations. What it's really good for is identifying materials with high breeding value. It's a way of using information from relatives to select potential parents more quickly. And that's the main application of genomic selection in increasing the rate of genetic gain. So we really need to be thinking about when we want to apply genomic selection, we don't need to do any cross-validation studies or measure the correlation between the GEBVs and phenotypic performance on the same set of clones anymore. That's not that won't give us a lot of information about how to use the technology. What we really need to do is structure our breeding programs so that they can make use of, so that we can select parents earlier and shorten the cycle. That's how GS is going to help us generate genetic gains. And so you've got a very nice system here where your F1C2 clones you're producing, and they're ready to evaluate in the 3rd year, if I understood your diagram correctly. That's where we need to be structuring our testing so that we identify potential parents out of that stage and the subsequent stage. So that you can get the breeding cycle down to 3 or 4 years. And the way we'll have to do that is take those clones and make sure that in every family we have a number of full sibs, that we have 15, 20 full sibs from each family, and then we disperse those around different locations in your network, not just 2 or 3 sites that you control directly. but with your partners in each of those major regions, and treat the full sibs as if they're replicants. So the genomic selection really allows us to substitute full sibs and half sibs for replicates, and how that's really important in clonal crop breeding, because it cuts down the number of years we need to increase those tuber progenies, so that we don't have to spend years building up tuber numbers in order to do multi-location testing. We can use the genomic relationship matrix to link information on families that are tested at different sites. This is referred to as sparse testing. So we, happy to link you with the genomic innovation quantitative geneticists who are working the system out in detail now, or discuss it with you further. But that's really where we're going to have the impact of genomic selection. Thanks. Sorry to go on so long.
Thank you,
Thank
Gary.
you, Gary. Thank you, Gary. I had discussion with you, I think, earlier last year also, and we are trying to do that, the sparse testing, which you suggested, and even Eduardo also suggested us to do the sparse testing, and we are working on that actually. So this is just the preliminary data which was generated. So just to see how it's working, we did this. You have already suggested us that this is not the right way to show that the prediction accuracies. So thank you, Gary.
Dorcas, can you unmute yourself, please?
Okay, yeah, thank you, Anna. Thank you, Salesh, for the nice presentation. And I do agree with all the comments that Gary just made. I just wanted to know, I have different questions, but just as follow-up on that, with the F1C2 clones, to what extent is this tuber multiplication still a problem at that stage to go for the even sparse testing, multi-location trials?
Yeah, Dorcas, actually, if you see in C1 generation, we hardly have 7-8 tubers. Okay, so in C2 from 8, the multiplication rate is, the maximum multiplication rate is 8 tubers from 1 tuber. So we'll get 8 into 8, 64 tubers. So if we have to distribute those tubers for sparse testing, I think we need one more round of multiplication.
Okay.
So it will go into the C3 generation, as Gary also told. So rather than in C2, we can go for the sparse testing in C3 generation, third clonal generation.
Okay. So my first question now is on the traits you showed about genetic gains, the 3 traits, I think, tuber weight and another one, and dry matter. And in most cases, your dry matter genetic gain was mostly negative.
No.
I wanted to know, is this as an essential trait in your market segment that needs to be improved, dry matter?
Yeah, thanks, Dorcas. It's a nice question, actually. And hopefully I didn't, I think I forgot to explain during my presentation. What happened actually in India, the focus initially was to increase the yield rather than the quality. So all the breeders, they went for increasing the yield, and they didn't bother about the dry matter, actually, which is a quality trait. So when we realized that by increasing the yield, we are decreasing the dry matter content in our varieties. So when we checked this, our latest varieties had low dry matter content in comparison to our older varieties. So that is the reason that we got low genetic gain for dry matter in comparison to other traits.
Okay, yeah.
But it's an essential trait now. It's an essential trait.
Okay. So
Yeah.
I'm asking that in relation to the breeding scheme that you have just shown, and also the GS studies that you had. As Gary mentioned, this is mainly for selection of parents. But in all those traits, again, I didn't see dry matter in the traits that you had. So I thought maybe that's one of the problems, because it's not being selected for in the program. And maybe that's something that needs to be changed. I was also wondering how or which selection index do you have, if in fact this is also an essential trait, and if it's being included with the other traits, what selection index the breeding teams are using. And then in the breeding scheme that you did show, even though you showed us enough cusp markers, I didn't see, or maybe this skipped my mind, where you plan to use the cusp markers in the breeding scheme.
Yeah, thanks, Dorcas. Actually, in potato, the number of markers are quite less. If you see the genes which have been mapped, or the markers which are available, mostly the markers which are available for biotic stress traits, and for other traits, for dry matter or the chipping quality traits, or for other traits, there are no robust markers available. So that's why we didn't include, although we have done some GWAS studies, we identified some of the SNPs, but we have not yet converted them into cusp markers unless they are validated.
Okay,
And
so the idea is to go forward with genomic selection, especially to reduce cycle length. Is that the plan that the team have?
Yeah. Actually, the plan is to, as I showed in my scheme, that for the conventional breeding program, it takes around 13 to 14 years. And we are just going with the same crosses, and those crosses are forwarded. So there is no reshuffling of genes, or no recombination is happening again. But with the genomic selection, when you take back your parents from F1C2 or F1C3 generation back into your crossing block with desirable alleles, so then your chances of getting gain for that particular trait or for index of traits is higher. So that is our idea.
Okay, sorry, and just one final comment. I'm sorry for going on. Okay,
No,
so
okay.
If this was to be the intention to apply genomic selection, then I think related to Gary's point, I think the program needs to look into, of course, overcoming the challenges of multiplying the tubers at F1C2, which will come in handy to reduce the cycle length as well, and also to see how to include all essential traits that need to be improved into the selection index, and which selection index they will use. Of course, you might know that we're recommending the desired gain index. And of course, to then update the breeding scheme to incorporate all this, including the evaluation strategy that Gary just talked about. Thank you.
Yeah, thank you. Thank you, Douglas. All your comments are well taken, and I agree with your points and Gary's points. And we are trying to actually relook our breeding program, because these are the things which are going on for so long, and changing the things, it takes some time actually to change the programs.
Thank you, Dorcas. Mokhtar, can you unmute yourself and ask to Salesh directly?
Yes, am I unmuted?
Yeah, Mokhtar, I can hear you. Go on.
Yes, so I was saying thank you, Hannah, and thank you, Salesh, for this nice presentation. And it's really good to see people using this dot tag marker, which is very promising. I had so many questions, but after Gary's comment and also Dorcas' comments, I think now I do have less. Maybe just a couple of comments and questions on the marker, like the different marker sets, starting with the cusp. Just to comment about what Dorcas was saying about when to apply or when to use the cusp markers if you go into genomic selection. I think here, for example, at what we plan to do is using the genomic selection as exactly Gary had explained, doing sparse testing, but we also have, as an information, some trade markers into the dot tag panel. So for example, for the PVY markers for this version 1 panel, we have PVY markers included into the panel. So for someone who is breeding for PVY, it's very convenient to use that version 1 panel to select for that trait. But if, for example, you are selecting for late blight resistance, which is not yet in the dot tag panel, mainly, for example, if you work with the R8 gene, you will need in the the next stages to select for late blight. And you know, sometimes you don't have the disease on your field test, so it's also convenient to apply those cast markers on a very reduced set of your breeding material. My question would be, for example, you presented several cast markers for the same trait, and generally they are in the same genomic region. So have you seen that some markers are more effective to predict the phenotype than others, and rely more on those markers and not trying to take the information from those that are maybe far away from the gene? So sometimes you may have some recombination between the marker and the gene. That would be my first question, then I can follow up afterwards with the dot tag markers.
Okay, thanks, Mokhtar. Yeah, we evaluated, actually, we developed other markers, the markers which Henels or you have developed at SAPE. So we also tested those markers also, and we developed our own cast species for late blight. And we have yet validated the 9 cast species which I've shown in my slide. And out of those 9 cast species, the results are not that good for all the traits, actually. With the 1 cast species, the number of accessions which are coming are resistant with 1 cast species are not resistant with the other cast species. So those are differing. So that, but yeah, the number of susceptible accessions, if we see with a favorable allele, there are no susceptible accessions. So that's why I told for preliminary selection, we can use these cast species. As far as the other cast species are concerned, which we have developed for late blight or other traits, we are yet to validate those, because the genes from which those cast species have been developed, or from the GWAS studies, we are yet to actually generate the data on that particular number of accessions. Actually, those are not phenotyped yet. So we are generating the phenotype data. So validation part is still to be done for those cast species. So that's why I told that only 20 out of those cast species have been validated with the phenotype data.
Okay, thank you. My next comment and question is about the dot tag panel. The dot tag panel has 2,503 markers. I saw you presented 1 marker less, but there is 2,503, and 2 of them are the trade markers I was mentioning before. And now there is a second version of the dot tag panel, which is now about 2,166 markers, and including 20 trade markers now. And the marker panel is currently available at DART. Maybe we just need I just need to send to another final marker file. So maybe it can be activated, let's say, into the system, so people can use it. And the specificity of the dot tag panel is that we have removed some markers that were of bad quality, and that will be where my question will come, is about your marker filtering, given the predictive ability you had. During the design of the marker panel, we have seen that there was some differences between the tetraploid calling if you look at the data data and if you do infinium array. So some of the markers had a very low, if you do the dosage calling, there is a probability that's called posterior probability of correctly calling the tetraploid dosage. And sometimes if the marker has a very low read depth, or if it is of what we call the bad markers, so you would have a difference between what you have in the dot tag and what you would have using an infinium array. So including those markers into your GRM may sometimes may reduce actually your predictive ability. So it would be, my question was, did you consider filtering those markers with very low, like with allelic bias, let's say?
Yeah, I showed in my presentation that initially we removed those markers where the read depth was less than 10. And then during the analysis also, some of the markers, as you told, that the genotype calling, when we saw the genotype calling, so they were calling only one type of class for all the accessions. Suppose for a one particular loci, we are getting only the like one kind of homozygote. Suppose 0 or 4, that is same across the accession. So we removed those SNPs also, and then we used for the prediction. But I think we removed only, I think 60 I wrote there. So out of this 2,502 SNPs, we used around 2,400 SNPs for our prediction analysis.
Perfect, perfect. That's very fine. Thank you. Thank you very much. Nice work.
Thank you, Mokhtar. So for the last question, Salej, Rajnish, are you able to unmute yourself and ask directly the question? Otherwise, Salej, I can ask.
Can you hear me now?
Yes, yes.
Yes. Hi, Salej. Thank you very much for nice presentation. I was surprised there is no nutrition breeding in the potato. So I was looking, are you doing any on this, on like the starch content and some other things on this, or only just fever yield and disease resistance on and the abiotic stress?
No, we have actually program on biofortification and nutritional evaluation also. But just to show this actually presentation was mainly for the genomics-led breeding and genetic gain. So for the scarce species and genomic selection, we are right now focusing on the processing quality traits, that especially in processing quality also the dry matter and the chipping quality. So the traits which are telling the starch content and all, they are being taken care in other programs like the amylose content alteration or the amylopectin alteration. So they are being taken care through genome editing or other approaches. But in this program in genomic selection and genetic gain, right now we have not considered that.
Okay, thank you very much. Yeah. Thanks again for lovely presentation.
Thank you, Thank you, Salej. So we came to the end of the 5th seminar of this series. Thank you very much to all for being with us, and hope to see you all in our next seminar. Thank you, Salej, for the great presentation. Thank you, Lenin, for helping us in this seminar. In the next 6th seminar, we will have Patrick Odong from IITA, Nigeria, to update us on the recent research on cowpea, and he will be introduced by Dr. Tim Close from US. So see you next time, and thank you very much. Thank you, Salej.
Thank you. Thank you, Anna. Thank you. Thank you, everybody.